I'm Dick Moberg, and for more than 40 years I've been developing technology to advance our understanding of the injured brain. I've had a chance to work with some of the leading minds in the field of neuromonitoring, including physicians, researchers, and entrepreneurs. I want to share their stories with you in the form of a weekly podcast so you can stay current on the latest developments in the field and the innovative people behind them. This is my neural network. Hi, I'm Dick Moberg, and I'm here in Berlin with Professor Jens Dreier, and he's at Charité, which is the top medical school in Germany. And he goes way back as one of the founders and pioneers of spreading depolarization research. Welcome to the podcast.
Yeah, hello, Dick.
Thanks, yeah. And we've had a lot of fun together in meetings and stuff.
Absolutely, yeah. We've known each other for many years.
Yeah, and certainly everybody I talk to looks up to you as really one of the pioneers in this new and exciting field. And one of the questions is, how did you get into this?
Yeah, actually a long time ago. It was in 1993. And it was by pure coincidence. So I had started in Munich to do animal experiments about functional activation. And my task was to test the influence of potassium and nitric oxide on functional activation. Functional activation happens when you move your hand and then blood flow increases in the brain. And I placed drugs on the brain surface of a rat. And all of a sudden, I observed funny drops in blood flow that could last for minutes and then recover. And I had no clue what it was. And it took me about 1.5 years to find out that this was an inverse neurovascular response to a phenomenon that at the time we called spreading depression. Today we call it spreading depolarization. So by coincidence, I discovered this phenomenon. It was published then only in 1998, 5 years later, but this was actually the start for me into this field. So I came from animal experiments.
And I think some of us know the history after that. So why don't you tell that about what happened with Tony Strong and with all that? So, yeah.
Yeah, we had started in the late 90s. We wanted to do a non-invasive project with near-infrared spectroscopy to measure these spreading waves in the brain, which actually turned out to be very difficult, and we never measured anything that looked like it. But we got in contact with a lot of researchers around us, and among them were in particular Martin Lauritsen and Martin Fabricius, and they were in close contact with Tony Strong. And Tony had the fantastic idea to place an electrode strip in patients with traumatic brain injury to search for these spreading waves. Martin Lauritsen and Martin Fabricius were part of this project, or became part of this project, which resulted in the paper in 2002. And then in 2003, this whole group of enthusiasts were meeting in Copenhagen to establish what we call the COSPIT group today.
Yeah, and that group has grown significantly over the years, and we've had, we've gone to quite a few of the meetings, and it's really exciting, something very new and fresh. And I think that, and the next meeting's coming up in Lyon.
Exactly, yeah.
And a little plug for that meeting. And if any of our listeners want to go to that, it's going to be a wonderful meeting.
Yeah, it will be a wonderful meeting. Yeah. Lyon is a beautiful city. The organizers, Stefan Marinescu and Baptiste Balanza, have done a very great work on spreading depolarizations in animals. And Baptiste is currently starting to establish this in the clinic. And we will meet from all corners of the world. Last time we were in Japan, and the meeting before was in Florida. So it's really international. And the great fun of these meetings is that it's very interdisciplinary. It's one of the few places where you can discuss basic scientists with clinicians, and they are all extremely interested in the work of the others.
Yeah, and we've enjoyed it for that reason. And in Florida, we Jed and I guess you have talked about spreading depolarizations as tsunamis, and so we brewed Brain Tsunami beer for the meeting in Florida, and we hope to have that beer again in Lyon.
Absolutely, and more T-shirts.
Right,
they're very nice.
Right, and I think that illustrates the community of this organization and the sort of closeness and the commonness of the purpose. And it's really to your credit and Jed's and a few others to really, you've made this meeting very special in terms of people looking at this common goal.
Yeah, I think that it's of course, I mean, what the background of all this is super serious because it's about people with stroke and traumatic brain injury. And we are trying to develop new approaches for better diagnostics and better treatment. But we also have fun, and friendship, and that is a good aspect.
Yeah. So why don't you go back to what happened after those the early publications? How has this progressed, and what do you think are the significant milestones?
Yeah, back in 1998, my idea was that this inverse neovascular coupling to spreading depolarization could play a role in subarachnoid hemorrhage for two obvious reasons. We needed two factors, high potassium and a factor that decreases the concentration of nitric oxide, either pharmacologically with a nitric oxide synthase inhibitor, or we could also use hemoglobin. So, and hemoglobin is very interesting because it is a powerful drug that lowers the nitric oxide level. It's basically not a drug, it's a protein of the body, and it is released in the subarachnoid space after subarachnoid hemorrhage. And we know that patients with subarachnoid hemorrhage develop delayed infarcts after the initial hemorrhage. This takes about a week, and then these infarcts occur. So our idea was that this could be the mechanism underlying the delayed ischemic stroke after subarachnoid hemorrhage. So the first step after we learned that we can measure these waves for me was that I wanted to know whether this occurs in subarachnoid hemorrhage. And this actually resulted then in 2006 in a brain paper where we showed for the first time that spreading depolarizations occur in patients with subarachnoid hemorrhage abundantly, a lot of them. We saw that in about 70% of the patients.
No, that's very significant. And it just was a year ago, there was a special edition of Journal Cerebral Blood Flow and Metabolism that I think you and Jed edited, which gathered papers together from a lot of other researchers. Maybe you can just talk about sort of the other research that is going on in this field, a little bit of a summary of what people are looking at. It's really spread.
Yeah. The phenomenon of spreading depolarization is the most underestimated mechanism of neurology. It is the mechanism of the cytotoxic edema that has been known to a few people for a very long time. It's now much more appreciated by a wide audience. So the applications of the spreading depolarization fields range from stroke to traumatic brain injury, migraine aura. They also have interactions in the epileptology field. So this is just the clinical field, and of course also circulatory arrest leads to spreading depolarization. Then it's very interdisciplinary. There are neurologists, of course, there are neurosurgeons. They implant the electrodes. There are radiologists. There are anesthetists. For example, Baptiste Balanza is from Lyon. He is an anesthetist. There are, from the basic science, neurobiologists. There are also modelers. I think the range of people that work in this field is simply enormous. And then, I mean, what are our goals? First of all, we would like to learn to read what's going on in our patients in the intensive care unit. Patients are often sedated and ventilated. You cannot do an MRI every half an hour or every hour, but you can do continuous electrocorticography. So when you see a cluster of spreading depolarizations, this tells you that this patient is developing a new stroke. Then you can start the treatment. But to determine the specificity, the sensitivity of all this, this is an important issue. Then the question of therapeutics: should we block spreading depolarizations, or should we not block spreading depolarizations? That's an important question. It's not so easy. It's clear that they are detrimental in areas where metabolism is disturbed, but maybe they also have beneficial effects in areas where they run through relatively normal tissue. This is not really understood. The blood flow response is a highly important target for the development of treatment. This is just a non-invasive development of recording that would also be an important goal, but also this goal is not so easy to achieve.
Right. And I was just going to get into that. And I think it's fair to say that we would know nothing about epilepsy if it weren't for EEG machines, right? And we've had those for many years. And I think that's one of the things that is probably holding this field back a little bit, just because it would be nice if you had a non-invasive way to detect these. And maybe you can talk a little bit about the challenges. I mean, as we talked earlier, that's sort of the holy grail. Why is it so difficult?
Yeah, actually, we published the first paper on non-invasive detection in 2012 in Brain. And we found very nice correlates between the subdural electrocorticography and the scalp electroencephalography. So when there was a spreading depolarization, this leads to a depression of the spontaneous activity. And you not only see that in the subdural electrocorticography, but you often see that also in the scalp electroencephalography. Unfortunately, the features are more difficult to interpret. For example, the spread is very well seen with the electrocorticography, where the electrodes are directly on the brain. But the spread is much harder to see, or it's not possible to see, when you do scalp electroencephalography. The slow potentials are giant when you do subdural electrocorticography, in particular when you do direct current or DC electrocorticography. But with scalp electroencephalography, these slow potentials are actually very, very small. So it is challenging to use that. There are also other reasons for depressions. For example, sedatives can also induce a depression of the activity. And to differentiate this from spreading depolarizations is a challenge.
Yeah, and that's going to be a tough one.
Yes, I think so.
I know you can use things like Laplacian transforms to try to focus what's underneath an electrode on the scalp, but I think even with that, it's tough just to see these patterns.
It also, plays a role that, of course, this is in the environment of an intensive care unit. Patients are washed, they are moved. So the electrodes have to stay in place. To have a good recording with scalp electroencephalography, it would be ideal to have a lot of electrodes. You have to take care that the electrodes don't press on the skin. All these practical issues have to be considered.
Well, I think we'll just have to wait.
We have to wait. Yeah. But there's also other technologies that may solve the problem, and we don't think of it now, or others already have it in their mind to develop this. Sure. We don't know.
Yeah. And I just want to relate. I was up at the University of Pennsylvania. This is about 2 years ago with my friend Ram Balu, and he was showing me a case in the the recordings of a patient in the ICU, and he had just become a big believer of spreading depolarizations, and he was he showed me, and they weren't even recording EEG in this patient, but he showed me these just big transitions in the metabolic measurements they were looking at, and some of the other parameters. And then he said, I wonder if those are spreading depolarizations. And he gave the patient ketamine and they disappeared. Now, this is just one example. It's an anecdotal case, but maybe there are other ways to detect this. So
far, I mean, there are many spreading depolarization is a phenomenon where basically everything changes. So it's huge. And there is all the ions, there is the changes in the transporters, in the transmitters. Every small molecule that is unevenly distributed between the intracellular and extracellular space changes during spreading depolarization. It's just the challenge to do it non-invasively. Invasively, for example, if you have an oxygen probe, then you can beautifully measure spreading depolarizations. You see that the transients, if you have the ECOG with it, you have a perfect recording setup, because then you also get information about the metabolism with the spreading depolarization. You see when the spreading depolarization occurs that very often you will have a dramatic drop in oxygen, and then hopefully a recovery. This tells you that the situation is still not too dangerous for the patient, but that also depends on whether the next one starts 5 minutes later. Then you should interfere. There's something going on.
Yeah, and it's I know that other places, I know here too, is when you start to look at these, they're in a incidence level that people never expected. And I know that Sharon Jewell's work with Tony Strong in London is at King's. They're seeing him in an incredibly high number of patients that they monitor. So, and I think Jed Hardings gave a nice plenary lecture at the Academy of Neurology meeting last year, calling this sort of the hidden culprit that you just can't see. So
yeah, it's actually what I find amazing is that it's kind of the culprit that you don't see, but on the other hand, it's a giant. So it's among all the brain waves that you can have, this is the by far biggest wave. So it's kind of paradoxical that on the one hand you don't see it, on the other hand it's the biggest thing you can imagine.
Yeah, and we can't see it.
We can't see it. Yes, with our electrodes. Now we see it, and we see it very often. We see it very often.
Yeah. So. Tell us about death.
Yeah, I mean, this is kind of when you do recordings in the intensive care unit, it's let's say part of the life of a doctor also is to usually we try to help the patient to survive, to recover, but sometimes patients develop complications and then they will die. And so during all these years where we do the neuromonitoring, we also recorded the death of the person during the recording period. And this actually was an eye-opener that we did this, published that, and I published this in the Annals of Neurology, because what we see is not surprising to us, but maybe surprising to others, that it's exactly the same sequence of events that you also see in other mammals. And which means after the circulatory arrest, you see the blood flow going down in the brain and the oxygen drops. And then after about 30 seconds, the brain activity stops. But this doesn't mean that the neurons are not functional anymore. You have a kind of a flat line when you do an alternate current electrocorticography. Then there is a silent period, And between 2 and 5 minutes thereafter, then the spreading depolarization starts, a terminal spreading depolarization as a giant wave. And what the question is, what happens during this silent period, and that we know from brain slice experiments or animal experiments. During this period, you have a release of small amounts of neurotransmitter, and they are more effective at the somas of the neurons, so they lead to an inhibition of the neurons. These neurons are not dead, although there is no heartbeat, there's no blood pressure, but the neurons are still alive and they are polarized. And then calcium slowly goes into these neurons and activates calcium-activated potassium channels. Potassium leaves the neurons. Then they start to depolarize slowly. And at the end of this period, then at one spot in the brain, this spreading depolarization wave starts. At the spot, it occurs within a few seconds, and then it spreads with a speed of about 3 millimeters per minute over the whole brain cortex, but also in other structures, thalamus, and also in the brain stem. So this means that the dying process takes much longer than you often read in medical textbooks. It's a process that takes minutes, and when people are clinically dead, they are still alive. When the spreading depolarization has started, you can still successfully resuscitate the person, because spreading depolarization does not mean the marker of death, but it is just the moment when the processes start that intoxicate the cells and eventually leads then to death. And the death occurs then over many more minutes. And all this is interesting. So when we published this case from the press, the greatest interest was with regard to near-death experiences, whether we can anything add to the discussion of near-death experiences. And I think, yes, we can, because it is often said that the near-death experiences, when somebody is resuscitated, occurs in a period when the brain is already completely in a state of lost function. But we know from the sequences that we see in brain slices in animals, and we now have also in humans, that in this period, you have still a very special type of neuronal function. And the interesting part, of course, is that it could well be that consciousness is connected with the polarization state of neurons. When you think about something, we believe that this has something to do with a short-lasting depolarization of neurons. There we have a process where billions of neurons within a period of a minute or so undergo a massive depolarization process. So nobody knows that, but if near-death experiences are real, then, of course, in my opinion, the pathophysiological correlate is are these processes first the transition with a silent period, and then the transition into spreading depolarization.
Oh, that's fascinating.
Yeah, I think what I find fascinating is that people that have near-death experiences describe things like that time actually is condensed, that they can be young and old, that they can be at the same time in different places. If we think that all our memories are somehow coded in the cells and all our neurons almost at once become active at the same time, then this is exactly what you would expect. I think the nice aspect of this is that reading more about these near-death experiences, almost all of them are good experiences, are positive experiences. They are kind of life-changing experiences. That's positive. So maybe there's a big concert in the end that we can actually look forward to.
Yeah, right, right. Well, speaking of that, where do you see this field going in the next, I don't know, 5 years, 10 years? I mean, where would you want it to go? I mean, we've talked about recording these non-invasively, but are there other issues with the science and the application of this to other areas?
I think the most important aspect of this is that this is very likely the most powerful mechanistic biomarker you can have to detect neurological deterioration in intensive care in these acute conditions, in particular subarachnoid hemorrhage, but also traumatic brain injury. So it's a biomarker. You can continuously record, you can use that to guide your treatment. And I believe this is the number one issue that is important. So it's important to compare this with the development of neuroimaging, to do serial neuroimaging and to see how it correlates with this. And then the next question is, of course, shall we treat it? And how shall we treat it? Is it good to block it or not? This is maybe the first question that we have to address. Or let's say the first question is also to establish approaches how to do such studies, because it's a very new field. I mean, we have to make it feasible to use this information for the treatment of the patients. That is challenging, and that is a very important issue. The block of spreading depolarization, that is one possible approach. And I think, in my personal opinion, the approach that is the most interesting is to modify the blood flow responses. Because when spreading depolarization leads to an additional ischemia through this inverse coupling process, through the spreading ischemia, then it produces additional damage.
Well, any further thoughts on, should we wait till Lyon to hear the latest?
I think that actually I'm really optimistic. I see that this field moves forward in the right direction, and it's really great to have more and more intensive care units in the world doing this neuromonitoring.
Yeah, let's hope that's the case, and good luck to you and your research. And I wanted to say in closing, I love your new office here with these beautiful tall ceilings, these windows looking out onto some very cool old brick architecture of the school here. So thank you so much for doing this interview.
Yep. So, thank you.
Thanks. So thanks for listening to this week's podcast. If you enjoy these interviews, please take a moment to rate and review this show on your podcast app of choice. Subscribe to Dick Moberg's Neural Network to receive notifications when future installments are available. Thanks for tuning in, and we hope you'll join us again soon.